including all the HIV plus TB and the TB without HIV. It's 0.55 thousand, which means 550 cases per, at that year, the year 2013. The prevalence, it's 22,000. The incidence in this year, it's 13,000. The incidence of HIV plus TB, it's low in Egypt because the HIV, it's very low in Egypt. It's only 97 cases this year. But the case detection in all forms, it's 59%, so about 41% are non-detected. In our study, we are concentrating on the immune response towards TB. Various T cells, macrophages, and also cytokines, they are all involved in the immunopathogenesis of TB. A better understanding of immunology of TB can not only lead to discovery of new immunodiagnostic tools, but also can help assist in helping in therapy, and suggesting new treatment regimens. In our study, the tool we used for determining the immune response towards TB was flow cytometry. Flow cytometry has hugely advanced the field of tuberculosis across all areas of research, using it in diagnosis and understanding the immune response towards TB. The aim of our study was to evaluate a panel of lymphocyte subsets, including CD4 positive cells, CD8, T lymphocytes, P lymphocytes, natural killer cells, T regulatory cells, and monocytes. After evaluation of these subsets of leukocyte subsets in active pulmonary TB patient, we also divided the diseased groups into 3 severity groups and determined these cells in the different severity groups. That was a case-controlled study of a sample size of 50 active pulmonary TB patients and 30 controls, age and sex-matched controls. We included active pulmonary TB patients who were diagnosed on the basis of clinical basis, radiological, and in the microbiology lab by positive Zell-Nelson staining, midget system positive, which is Proth automated culture, and a chest radiograph, as we said. Both male and female were included, and consenting to the experimental lab procedures were taken. We excluded HIV positivity, diabetes mellitus, immunological or autoimmune disease, and pregnancy. The control subjects, they were 30 healthy controls, had no abnormal chest X-ray findings, no underlying disease, and of course, consenting to the experimental lab procedure. The method, the active pulmonary TB patient, where they had clinically assessed, radiologically examined, and microbiologically. Microbiology included Zell-Nelson staining, tuberculin testing, and midget TB culture. And after that, the hematology, after being diagnosed as TB positive, the hematological investigations in the form of complete blood picture, ESR, and flow cytometry for the panel of the leukocytes, which are CD3, CD4, CD8, CD19, CD16 plus 56, CD4, and CD25. Of course, CD3, the T lymphocytes, and they are divided into CD4 and CD8, which are T helper and T suppressor, CD19, the B lymphocytes, CD16 plus 56 are the natural killers, 425 are the T regulatory cells, and monocytes, which are CD14. The healthy controls, we have done to them tuberculin testing to be sure they are negative. Hematological examination, the same, complete blood picture, ESR, and flow cytometry. The active pulmonary TB patients were divided into 3 groups of severity according to the X-ray findings. The first group is the mild cases involving single loop without visible cavities. The second group, it involved unilateral involvement, but 2 or more loops were involved. The third group, there was massive involvement, bilateral. The microbiological examination revealed in the active pulmonary TB patient, they were positive by Zell-Nelson staining, positive tuberculin testing, and positive for the Bactec Midget 960 TB culture system. The entire healthy group had negative tuberculin test. So these are the results. First, the hemoglobin percent. That's the significant results, statistically significant result. There were lower hemoglobin level in the active pulmonary TB, significant higher mean neutrophil percent, and significant lower mean lymphocyte percent, and significant higher ESR. That were the conclusion of the result, the most significant. So that's the hemoglobin level in males and females. Of course, it's lower in the TB patients. The neutrophil, as we said, neutrophil percent were higher in the TB patient, and the lymphocyte percent were lower in the TB patient. Regarding the lymphocyte subpopulation, we've first the absolute number one. That's the absolute leukocytic count. That were higher in the active. We will discuss the active pulmonary TB. They had lower lymphocytic count, absolute lymphocytic count. CD3 were higher. CD4, which are the T helper cell, were lower, but CD8 were higher. That made a CD4 CD8 ratio significantly lower in the active pulmonary TB patient. That's the B lymphocytes. They were significantly lower in the active pulmonary TB. The natural killer cells were higher, but that wasn't significant, and CD14 monocytes were lower, and the T regulatory cells were higher, but the significant was the B lymphocytes, which were lower at the active pulmonary TB. That's the box plot for the CD4 CD8 ratio being lower in the cases, and the B lymphocytes being lower in the cases. So that's some of the literature. That's some studies. The first study revealed that CD4 CD8 ratio is considered to be the index of cell-mediated immunity, and TB. That in our result, it was lowered in the active pulmonary TB patient. So TB is a reversible cause of lowering CD4 CD8 ratio. But regarding the B lymphocytes, up to date, the contribution of humoral immunity regarding B lymphocytes and the immunoglobulin response in TB is still largely unknown. That's regarding the disease severity groups. There are 3 disease severity groups: advanced, moderate, and mild. The most significant result we found that the T regulatory cells. We found they are highest percent of the T regulatory cells in the active pulmonary TB patient, and lowest in the mild group. Highest in the advanced group, and lowest in the mild group. That's a graph. So the regulatory T cell, that's also some studies reveal that regulatory T cells prevent eradication of tubercle bacilli by suppressing efficient CD4. The T regulatory cells, it dampens the immune response by suppressing CD4, which are the T helper cell. So that's why in the advanced cases, TB patients with advanced cases had large amount of Treg cells. The T regulatory cells also tightly correlates with disease severity. Other studies say that they correlate tightly with disease severity, and they decline after successful therapy. Other studies reveal that attenuation of T regulatory cells along with BCG vaccination has a positive impact on the protective immunity against TB. To conclude our results, we found that CD4 T cell percent were profoundly decreased, and CD8 were profoundly increased in active pulmonary TB patient, leading to a significant decrease in the ratio, CD4 CD8 ratio. And not only the CD4 lymphocytes, which were lowered, but also the B lymphocytes, and that suggests the unappreciated role for the B lymphocytes in the immunity towards TB. We reported significant high percent of T regulatory cells in the advanced group. And we suggest that this marker can be utilized as an indicator of disease severity and prognosis with the treatment after that. A clear picture of the network of immune response to the pathogen, as well as understanding of their effector function, is essential to design and implement effective vaccine and treatment for TB. we had some recommendation. It's recommended to combine phenotypic and functional analysis of T cells and B cell subset, not only in peripheral blood, but also in bronchoalveolar lavage. If we combine both in blood and also in bronchoalveolar lavage to know the local immunity inside the lung, that will be of more benefit. It will be of value to measure these lymphocyte subsets in the newly diagnosed TB patient, and follow-up studies are required to determine the change in these subsets, what will happen with treatment. So when they are newly diagnosed, we will determine them, and then after that with therapy, we will determine have them been increased, decreased, or what happened. The exact protective role of B lymphocytes is undetermined, so it needs to be thoroughly investigated. The T regulatory cells especially has an important role in dampening the effector immune response to TB infection. So these cells should be evaluated, especially in the newly diagnosed, and after that for the prognosis, to evaluate their potential role in immunotherapy and vaccine designing. That's acknowledgement, and these are the references for my study, and thank you.
Thank you. Okay.